Andate a casa, non c'è nulla da fare. 25 anni fa, una diagnosi di distrofia muscolare di Duchenne.
È vero che i miei muscoli diventano sempre più deboli.
Una patologia degenerativa rara che colpisce i muscoli. Ma c'è chi non si arrende e fonda Parent Project. Intanto il mondo cambia e anche Parent Project cambia un po' il mondo.
Grazie a Parent Project e
approccio terapeutico.
L'ultimo anno finanziato da Parent Project.
Il project che ha
circa 8 anni della prima volta che in maniera.
I pazienti oggi hanno un futuro da immaginare, degli obiettivi da raggiungere e dei traguardi da conquistare. Oggi, anche con la Duchenne puoi diventare ciò che vuoi. Come
Luca Buccella, che ringrazio per essere con noi.
Correre insieme, in fin dei conti, è una sfida già vinta.
I'm here to tell the future is there.
Jacopo,
Alla Duchenne.
Matteo, Lorenzo, Luca, Samuele, Francesco e come tanti altri ragazzi che vivono una vita piena. Ma questo non basta, non c'è ancora una cura e tanti sono gli ostacoli da superare e le strade da aprire.
Tutti insieme
Abbiamo formato una grande famiglia.
Scrivi i prossimi 25 anni di storia insieme a noi.
Benvenuti e bentornati alla nostra 18ª conferenza internazionale. Siamo arrivati all'ultimo giorno.
Welcome. This is our last conference day. It's day 5. Actually, we've been going live since last Wednesday from our TV studios, so to speak. Well, actually, this is our headquarters, but as of tomorrow, this is what it's going to be, once again. We'll probably miss these live streaming conferences and sessions, but we have certainly missed being in person. So what has gone on so far is all very good, but we have missed you so much. I wish to thank all of you, those of you who are abroad and where you are, it's probably nighttime, so thank you even more. Anyway, as all other sessions, there will be simultaneous interpretation in this session too. 2 languages are available, Italian and English. To have access to translation, you'll just have to click on the language of interest, thus allowing our great interpreters, as the speaker and the interpreters thank her, who are really doing their best to allow us all to understand each other, both into Italian and into English. And by the way, we would like to stress once again that all speakers are kindly requested to speak slowly, thus allowing our interpreters to work as best as they can. Now, we're very nervous today because we're going to introduce a very special document, actually one of a kind, because there is nothing like that around the world. For that, we wish to thank Dr. Pegoraro. We can see her. She's always been on our side, speaking in a very clear way. She's so good that she can make everybody understand even the most complicated things. And then 2 of the 3 chairpersons of today are in their home, Bellon and Laurenzana, whereas Fernanda De Andres is here with us. Gerardo is in Potenza, central Italy. He is one of the board members of our association. In particular, he deals with the counseling center as well as all activities carried out together with the parents all across Italy. He is the father of Antonio, 18 years old, and there's also Alessia, who I believe is 15 years old, and his wife is Julia. It was 2009 when he first got in touch with Parent Project, and since 2017 he's been a member, as I said, of the Parent Project board after he had been working at a local level, namely in the central Italian region of Basilicata. And then Antonio, we're very happy to have you here. He's 28 years old. He's a BMD patient and a very affectionate member of our association. And of course, it goes both ways. He graduated in mathematics in Padova, and he is now on a PhD program dealing with artificial intelligence mainly. He's actually involved in several researches on applied maths in artificial intelligence. Well, congratulations, Antonio. And then next to me, Fernanda De Andres, who needs no introduction. She's one of the molecular biologists working in our scientific unit. She's been with us for the last 10 years, together with Ilaria and Gloria. She is responsible, as I said, for the scientific unit of our association. And she's been behind this great, excellent work that you'll be hearing about in a minute. So thank you all. And now the chair people have the floor. I don't know who's going to begin, but you have the floor. Fernanda will be starting. Thank you very much, Cristina. Indeed, I am very nervous this morning, precisely because we're going to introduce you to this document, which is really one in a kind. I mean, there's nothing like that worldwide. The first time actual guidelines on the clinical management of BMD for families. This is the first time that such document has ever been drafted. Thanks to many clinicians who have participated, but also and more specifically thanks to the great will and determination of patients. So it's a very rich work. We would have never been there, by the way, unless Dr. Pegoraro had contributed. She's here with us precisely to tell us about such guidelines. Anyway, there are many more people behind the document, hence the relevance of acknowledging them all. For example, my colleagues from the Duchenne Counseling Center, they have been responsible for the drafting of some parts of the document. Aurora, our graphic designer, she's so good. I don't know how she can do that. And then Elena and Fabiana, who have reviewed all the texts. They have really been editing the whole story. The document will be very soon uploaded in our website, but today, for the first time, you'll have the chance to download it, as you can see from clicking on this QR code. Of course, we would also like to thank all those who have helped Dr. Pegoraro drafting the document. Of course, the clinicians working with her, including Professor Comey, who will be taking the floor about givinostat later on, as well as Dr. Adorisio, Bello, Bertoli, Cicenia, Adele D'Amico, Grazia D'Angelo, Alessandra Ferlini, Alessandra Fortunato, Esther Giaquinto, Joascon, Fabrizio Racca, Andrea Vianello, and all those staff members. They have all supported and contributed to the drafting of this really outstanding document. Thanks for deep in our hearts. Families were badly in need of such document, and without you, we would have never made it. So, having said that, the floor goes to Gerardo for actually starting the session. Gerardo, you have the floor. Good morning. Thank you, Fernanda. I don't really know whether I will make it because I'm really very nervous and so very happy. I can still remember back in 2014, there was a webinar precisely devoted to BMD. Possibly it was the first time that was being the case. But then there was a technical problem. We had to stop for a few minutes. Dr. Pane could not, talk, but the chat was still active. And actually, many people started using it to write and share their feelings. So it seemed like for the very first time, there was especially, a special place for us to share our experiences. Hence, the relevance of such sharing a time that we seem to need more and more. So we have organized more BMD meetings within the framework of the 2018 and 2019 international conferences. It was precisely on those occasions that families asked for guidelines about the clinical management of our disease. And it is our extreme pleasure to be able to show them and introduce them and introduce the document to you. And actually, Antonio will now have the floor to go into the details. So good morning. Good morning, everybody. Thanks, Gerardo. Well, I'm very, very happy to participate in this beautiful international conference. Thanks, Bern Project, for organizing it. Of course, we must keep distant within this particular conference, but it's very nice to see that although only virtually, we still have a dedicated time for BMD. I believe this is of paramount importance, first of all, for patients and their families, but I believe also for physicians, researchers, and the companies, I presume, at least. So the one word that seems to me to recap the whole of the whole relevance of the international conference is sharing, and having a BMD window within this international conference allows us all to be connected in a network. And once again, I believe this is of paramount importance for us to cope with BMD as best as we can, possibly having further tools to not feel alone. Now, apparently this session is already successful, and that clearly speaks for how badly needed it was. So now we have a special room devoted to BMD, which is actually different from DMD, and here is the reason behind the project that has now at last been achieved. That is the document containing specific BMD clinical management guidelines. So I think that this is something that has allowed patients to be fully involved, and this is going to be even more and more the case because through this document we have just encouraged such attitude in patients, and hopefully this, as I said, will keep being the case. So these guidelines are not, a simple handbook. Actually, it contains several pages that can all be useful for certainly for those who have just received a BMD diagnosis, but also for those like me who have been living with BMD for quite a long time. And the topics range from clinical management to the management of the motor issue, as well as the psychosocial aspect, which I believe is as important. So having said that, let's start. And the floor now goes to Professor Elena Pegoraro, who needs no introduction, but I would still like to introduce her to you all. She's professor at the Neuroscience Department of the University of Padova, Northern Italy, and she is in charge of the neuromuscular unit within the same university. She's indeed one of the main points of reference for what pertains to the clinical management of both DMD and BMD, both in Italy and abroad. And she's played a key role in drafting the guidelines, namely insofar as she has been coordinating the work of all the drafters, all the authors of the guidelines. So we'd like to acknowledge her for that, also on my personal behalf, if you wish. So now you have the floor.
Professoressa Pegoraro, mi dicono che deve attivare l'audio.
Credo di averlo fatto, mi sentite?
Eccoci, ci sentiamo, sì, e la vediamo anche lo schermo.
Perfetto. Buongiorno a tutti, grazie per la...
Good morning, everybody. Thank you for the kind introduction, and I am extremely glad of seeing this document ready. It is necessary to underline that this is a collaborative work with the participation of all the clinicians that are listed here, and my co-workers as well. And it was, under the initiative of Duchenne Parent Project. And I wish to thank Fernanda, because over the years, she's been the one who has kept us motivated, and also she has managed the different clinicians who sometimes were not so keen on respecting guidelines. What these guidelines are not. They are not a self-diagnosis tool, and is not a tool for the self-prescription of drugs or tests. Well, if we look up Google for Becker muscular dystrophy, we have 752,000 entries. And so this excess of information makes it difficult for families to understand how much is correct and how much is not. Unfortunately, together with very appropriate information, we also find information which is not appropriate and correct at all. That is why we have imagined these guidelines that starts with diagnostic suspicion and confirmed diagnosis, and then a multidisciplinary approach. And you see how many specialists participate for an appropriate management of the patient to share ideas about the disease and the diagnostic and therapeutical route. I'd like to share with you, the most important message for each chapter. First of all, diagnosis. The clinical suspicion is based on the appearance of a deficit of strength, but also an isolated increase of CK, this muscular enzyme that shows that there is a muscle suffering, and that may lead the patient to consult a physician. And so when you have a patient with a Becker disease, you have to extend also the analysis and clinical observation to family members. Another aspect is the extreme clinical variability of BMD. We're speaking of one single disease, but this disease may have very severe forms, intermediate dystrophies, where there is an early loss of ambulation, and the most typical forms where ambulation is lost only in the adult age, or mild forms where ambulation is maintained, but there is a deficit of force. And there are also some diseases that are asymptomatic, where force and muscular function are preserved, but the patient may have other forms of myalgia. Once there is this suspicion, you must have the genetic confirmation of the mutation. Normally, it is a multi-step process. There is a first level of investigation, this MLPA. This is a lab method to identify the presence of large deletion of the gene, or and it covers essentially most of the variations in BMD. Of course, you have to read letter by letter the whole gene with sequencing methods, with a specific method of investigation, and then you reach a third level where well, you also have to do RNA analysis. We must remember that BMD is a rare pathology. This is something we must underline, because both at the level of diagnosis and after the diagnosis has been made, you can have access to a number of facilitations. So the certification of rare disease, and so you will have all the drugs and tests free of charge. So for a multidisciplinary management of the patient. So the diagnostic pathway that starts with diagnosis has a specific code, and all that can be done without paying anything. Everything is paid for by the National Health Service. Then there is this change in the life of the patient and the family when this diagnosis has been made. And and there are, of course, different psychological effects. The genetic counseling is also the time when you can discuss decisions for the future, a prenatal diagnosis, both with invasive techniques and non-invasive techniques, or pre-implant diagnosis that now can be carried out in some centers, again, free of charge. It's all paid for by the National Health Service. Once you have the diagnosis, let's say that the story starts. And there must be an alliance between the physician and the patient, because only if we build that alliance can we manage the disease and in a effective way. The neurologist, the neuropsychiatrist have two roles. On one side, they must monitor the muscle function, and the muscle function is assessed with the standard, with the classical standards, assessment of force, speed in carrying out some movements, and associated with emerging techniques such as NMR, where you can follow the evolution of the disease in the skeletal muscles. Why this is important? Because you can monitor over time the clinical changes of the patient to understand both the natural history of the disease, but also the efficacy of rehab, for example, and we hope very soon also the efficacy of a possible therapy. We are speaking of a multidisciplinary management. You must consider the good health of the bone. The bone is not an inert tissue. It is a tissue that is perfectly alive, that is very important for function and exercise, and at an early stage of development, it's also very important, but also in order to maintain a good calcium, good levels of calcium. So you may have a reduction of function, so a reduction of exercise, and that may have a negative effect on the bone. Some patients may have an ambulation that is quite unstable, which means that they are prone to falling, and keeping a good bone metabolism, of course, helps them, protects them against traumatic fractures. One small note, when we speak about fractures, well, it is, of course, a very special moment in the life of the patient, and it is important to take the most appropriate therapeutical decision. This must be shared. I mean, a common decision, orthopedician, neurologist, cardiologist, and so on, so that you can choose for each patient the most adequate treatment, because protracted immobility may lead to a loss of function. So all efforts must be made so as to make the patient move as soon as possible. And again, I stress the importance of motor activity for the trophism of the bone, but also for motor health of the patient. So rehab, neuromotor rehab is extremely important to maintain autonomy, delay the evolution of symptoms, and prevent complications. So if you want to do something in this, to solve this vicious circle, so there is a reduction of motor activity and modification of the bone metabolism and bone atrophy, increase of muscle weakness, and again, reduction of motor activity. So this is a vicious circle that must be modified thanks to rehab intervention. Of course, this rehab varies depending on the stage of the disease. So you must do stretching, for example, and also an adjustment, a modification of the house itself, of the home itself. So eliminating carpets, steps, surfaces that are not perfectly flat. And also the management of the chair is very important. And in rehab, robotic systems for environment control. We have one session specifically on that. Voice recognition to use PC, telephones, and whatever, cell phones. I mean, any sort of electronic system to guarantee good environment control. Certainly, the respiratory and heart function are of paramount importance. As for respiration, well, is not as, respiratory failure is not as common as in DMD, but it must be monitored. The early signs are fatigue, morning headache, difficulty at concentrating. And what are the tools? Classic tools, strength of respiratory muscles, lung volumes, FVC, and so, and FF1, and cough, of course, is a beneficial mechanism so as secretion do not stagnate and they don't become a plug for the development of bacteria. Then gas concentration in blood. Night monitoring is very important because through night monitoring you can identify the presence of respiratory failure, and mechanical, non-invasive mechanical ventilation and IV could be a solution. It is an important moment, but precisely to improve the quality of life and life expectancy, because that reduces respiratory complications. And then cuff support when necessary with abdominal compression or with the AMBU machine, and the cuff machine. Oxygen therapy is very important because it can correct the concentration of oxygen in the blood, but not the underlying pathology. The heart function that is very important in BMD. When we have a heart failure, advanced heart damage is extremely dangerous. So you need ECG, lab exams, Holter, and up to NMR, heart NMR. And in the guidelines, you have a list of the drugs that are used more frequently, and all that non-pharmacological therapy that is used for BMD, defibrillators, the ICD, VAD, and that is becoming increasingly accessible for BMD patients. Necessiology and sedation are also very important, because we must remember that in BMD, like any other neuromuscular disease, we must avoid some drugs that are associated with severe complications during anesthesia. So the anesthesiologist must be aware of the disease of the patient, and he must be aware of the guidelines for safe sedation. If there is important surgery, the cardiological and pneumological assessment must be made. And if the patient is under cortisone, which is more rare, but it is necessary also to increase the dose of glucocorticoids to avoid adrenal crisis. This is the emergency card that is very important. It is obvious that for a planned intervention, you can consult different specialists, but if the patient goes to an emergency unit, he must have this emergency document explaining what are the possible complications or procedures to be avoided. Then nutritional aspects. In a patient with reduced mobility, you can have weight gain, or in non-deambulating patients, you may have the opposite phenomenon, so the reduction of lean mass. It is very important to be aware of the calculation of the percentage of fat mass and lean mass in the body, and gastrointestinal problems, because sometimes you may have a slowing down of GI transit. Then psychological aspects, last but not least, because once the patient receives the diagnosis, he must learn how to coexist with BMD, and the coexistence with this disease must be proactive, and because these patients can have a very full life and they can achieve all the goals they want to. It is very important here to have psychological support that must be adjusted depending on the age and the phase of the disease. Then social, administrative aspects, work aspects. Well, there are some facilitations given by the law when you have a given disease or a disability. So promoting independence, autonomy, and so on, but also being aware of the legislative aspects whereby the patient can have access to a number of services and home care. Of course, that was a very rapid overview of these guidelines. And I close by stressing once again that these guidelines have been the result of a big cooperation among all the clinicians who have participated, depending on their skills and I mean, it is something that Parent Project was much insisted on. And why I'd like to go back to what Antonio wrote. It is important to get an immediate diagnosis and appropriate management to be able to face the new, to consider the new therapies that are emerging, having the function preserved as much as possible. These guidelines provide the patient and the family with a very summarized and simple tool, but very accurate tool to get information about the different aspects of the pathology and to navigate on the net with the ability to understand what is correct and what is not among all that is published on the net. Thank you for your attention.
Prima di passare la parola a Gerardo per
Thank you very
introdurre.
much, Elena. Before giving the floor to Antonio for him to introduce our next speaker, I'd like to remind you all that you can just ask your questions as of now, just using the Q&A part of the Zoom screen. So just write down your questions there, and then all questions will be answered at the end of the session. And now the floor goes to Gerardo, right? Okay, for introducing us to the next speaker, who's going to tell us about some new clinical trials in BMD, as well as the other speakers who will follow. I'd like to thank Professor Pegoraro. Let me say once again that you can download this document just visiting the website of our international conference. Now, the floor goes to Professor Giacomo Comi. He is a professor at the University of Milan and the director of the neurology unit of the Cagranda Policlinico in Milan. And he is also a major reference for those of us who deal with BMD. And he's been involved in several clinical trials for quite some time. I'm thinking in particular of the givinostat phase 2 trial. Actually, not many clinical trials consider the possible treatments for BMD patients, but the study that Professor Comi will revolve around is indeed the study number 1 having ever been carried out in Italy for our disease. So it is indeed a pleasure for us to now listen to Professor Comi. You have the floor. Good morning. Good morning. Good morning to you all. Thank you very much for this introduction, and I'd like to thank Parent Project for organizing this international conference which deals with so many topics in such an accurate way. Now, my contribution will be a very simple one. Of course, I owe a lot to Professor Pegoraro for her wide-ranging introduction. So I will just tell you about the state-of-the-art of the givinostat study for BMD. Elena has already gone through the different aspects characterizing BMD, so I won't go into the details, but to say that a major issue is variability when it comes to BMD. You know, onset timing may change, phenotypes may change, although they are milder, but certainly give rise to muscle pain. So, I mean, it is a kind of a disease that actually includes a wide range of clinical manifestations. And such a clinical heterogeneity also entails the current lack of proper pharmacological measures, not to mention the lack of studies and clinical trials. We really have very few of them. So the givinostat is an HDAC inhibitor, as we know, and the study with this product is now just one of the very few studies being carried out on BMD around the world. Hopefully, new potential treatments for BMD will come out of such studies, and I do believe it's just going to be a question of how soon or how sooner or later this will be the case. So, what happens to dystrophin in BMD? Well, first of all, we must say that dystrophin makes the muscle membrane stronger, thus making it more enduring and resistant to whatever happens during contractions. However, dystrophin is also a molecule that has to do with different muscle cell signals, and within the muscular tissue, it also plays a role. So let's consider such signals which sometimes imply an impairment of several genes, actually a whole cascade of them. So doing something about gene regulation is indeed one of the things that can be done downstream the either partial or total dystrophin deficit in order to hopefully do something about regeneration or about the fibrosis mechanisms that occur within the dystrophic muscle. So, having said this, and based on the experience we have all had in the field of DMD, together with the people from Italfarmaco, we have designed this study, which is mainly a pilot study. It's a phase 2 study, controlled, and givinostat is within a double-blind study design. Well, approximately 48 patients were supposed to be eligible for randomization and treatment for a period of 12 months. As for the administration of either givinostat or a placebo was supposedly twice daily. The first patient was enrolled in February 2018, the last one in November 2019, but in the middle, in 2020, something happened, which is called COVID-19. So, of course, this has changed the whole framework. Therefore, the very last biopsy will be carried out on February the 25th, 2021. Then a 30-day follow-up will follow. Two Italian centers have participated. One is the one where I work, and actually there's another one which is not Italian, in Leiden in the Netherlands. So of course, as for the inclusion criteria, the BMD diagnosis was to have been genetically confirmed. This is the age range from 18 to 65 years, and then the various functional tests were to show some specific scores. Here you see in particular the distance to be covered with the 6-minute walking test from 200 to 405 meters. Then no heart disease was supposed to be there, no cardiomyopathy rather. As for heart failure, only some neoclasses were admitted, and the ejection fraction was supposed to be over 50%. As for drug therapies, well, maybe steroids or ACE inhibitors, this was accepted. As for platelet values, they were to be within the normal range. The aims of the study, well, first of all, to establish the histological effects of givinostat on the biology of the dystrophic muscle as compared to placebo. The focus was mainly on some specific disruptions such as fibrosis, which is a major characteristic of all muscular dystrophies, I might add. And then other endpoints were also included, so-called secondary endpoints, namely to establish the macroscopic muscle effects, mainly using MRI and also so spectroscopy. And then we wanted to establish the other histological parameters within the muscle, and also to establish the efficacy of givinostat based to a certain number of functional parameters that had been acquired in the meantime. But our attention was always focused all through to safety and tolerability of givinostat as compared to placebo. In the adult population, we seem to have acquired the most interesting data. We have tried to go deep into these patients' quality of life, but also other aims included were mainly compatible with the study being a pilot study. So here you see the actual number of patients who have been enrolled. Some only participated in the screening without actually being enrolled in the trial for the lack of one or more inclusion criteria. Otherwise, you see the age range and a remarkable time gap since diagnosis, that is 13 years. As you can see in genetic terms, over 94% of patients showed deletion, mainly due to the inclusion criteria that we had drafted. As for other characteristics, the ejection fraction was over 61% as according to our inclusion criteria. One of the secondary endpoints, as I said, was about function tests, the 6-minute walking test, as I have said, but we also asked patients to provide us with data obtained, for example, with the motor function measure, basically measuring some important elements such as the domain 1, so to speak. We also got some data regarding the 4-step walking and 4-stairs climb and the walk, run, or rise from floor. And myometrics was also used together, of course, with the QOL questionnaires. The muscular biopsy has been of paramount importance, and actually the biopsy, as I said, was a major parameter here, namely in terms of the rate of fibrosis in black on the bottom side of the slide. However, sometimes sometimes we got different histology frameworks. You can see this from the different pictures. As for muscular regeneration, it was also different from time to time. And let's actually talk about regeneration. This is immunohistochemistry with antibodies of myosin heavy chain. Now, this is about the quantitative data we obtained from the Italian cohort of this study, including 45 patients that, if compared to control age-matched groups, the fiber size variability changes. And you can see the same for nuclear centralizations or the portion of the muscular fibers that undergo significant changes, and also the number of the splitting fibers. As for necrosis fibers, well, here differences were not that many, which accounts for the chronic nature of the disease. Anyway, by measuring the different phenomena, the quantitative data can actually be very, very accurate and precise at the end of the study. This is better shown. You see every patient is seen as to the quantity of fibrous tissue in yellow. You see there's a sort of a gradient, cross-cutting the patients. And you can see that there are also other elements such as the fat tissue or other histological structures. Now, fiber size variability, it is indeed a major issue for BMD as it emerges from this study. You can see it here. It's around some values for other for the control group, not so for BMD patients. You can see we range from hypertrophy to hypertrophy quite often, and indeed the variability score is very, very high. And again, let me stress how relevant this piece of data, as it has been constantly and accurately shown by this study. Now, these histological parameters correlate, of course, with each other, so to speak. All positive correlations are here in yellow, colored in yellow. As for total fibrosis, well, it shows a negative correlation. It's a statistically significant score concerning the portion of the muscular tissue that is involved. However, this is not the only aspect because there is also a correlation between such histological aspects and the functional tests. So, as for, the various TFTs for the functional tests, well, they have shown such correlations. The same goes for the force that is measured, including the one exercised during a limb flexion. Now, this is to show the same thing. Basically, you see that parameters may correlate either positively or negatively, as I have already said. And this is another way of seeing that through a graph. You see that there is a total fibrosis and then the brachial biceps myometry, which is the one about the grip strength that I mentioned before. Now, I have already said that another parameter is RMI to study the macroscopic histology of the muscle, but also to consider the muscle within a wider ranging pharmacological approach to come. So, among the different parameters acquired with RMI, and in particular with the MRS, is the fat tissue and the fibroadipose tissue in the pelvic girdle and in all lower limbs. Now, when then one considers such correlations, well, histological parameters more specifically correlate with function tests, as we have said, but also significantly so with the MRI data. Here you can see, for example, the MRI regarding the fat fraction and the fibrosis that had been seen histologically. So there is a correlation, there is an obvious, I might add, correlation, and this goes for MRI and also for MRS, as we have said, especially regarding the correlation with the parameters acquired through more conventional methods. MRI and the test functions also correlate. So function tests and histology, but also MRI and function tests. Here you see a graph showing the correlation through MRI between the fat fraction in the thigh muscle and the Forstairs test, or else there's again a correlation in quantitative terms through MRI or MRS between the D1 scale, the domain 1 scale, and the soleus. Okay, that was about the soleus. Now, some adverse events have been detected, mainly regarding platelets. There was a reduction in the platelets of almost all patients. And of course, this also includes, this list of adverse events also includes the excess in triglycerides triglycerides, which was seen in 2/3 of the patients. So, as I said, the last biopsy in the study will be carried out in a few days. Therefore, we'll be able to compare our study with the other longitudinal analyses that will lead to the results being available in the second half of 2021. Let me conclude by thanking all the colleagues, both in Italy and abroad, who have all cooperated. Thank you very much. Thank you. I'll be happy to answer your questions, of course.
Grazie mille, do direttamente la parola a Antonio.
Thank you, and now I give the floor to Antonio. Thank you, Professor Comi, for the line of reasoning and also for contributing in the guidelines. And now we give the floor to Ransi Somaratne, who is the medical director of Epirion Bio. He'll be telling us about the EPM01 study, phase 1. And the target of the study is the mitochondrion, that is a fundamental element for our cells to produce energy. They absolutely need it to carry out their activity. Thank you.
Okay, hopefully you can see my screen. Great, thank you.
Ok, ora ok.
Okay, great, thank you. Okay, thank you very much. As you know, we have a phase 1 trial of EPM01 underway in Becker muscular dystrophy. We're pleased to note that we have been able to close screening, and we should be fully enrolled very soon. We have approximately 20 to 22 patients in the study. This last year, we received FDA and EMA orphan designation for this drug, EPM01, which is a form of plus epicatechin. In this study, this is an open label dose escalation trial. We're testing 3 doses. Each patient will step through all 3 doses of 75, 150, and 225 milligrams of EPM01, which is given by mouth every day. All of the patients in the study have genotypic confirmation of Becker or Becker-like muscular dystrophy, and we will dose escalate every 8 weeks. We're conducting muscle biopsies at baseline at the highest dose level at 6 months, and then again at 1 year. We're doing cardiac MRI at the same time points. In addition, we are looking at plasma biomarkers, which not only tell us about the mechanism of action of the drug, we're also looking at plasma biomarkers that are associated with the underlying disease, as well as 6-minute walk test, time to run, walk 10 and 100 meters in the 4-stair climb, and an assessment of hopefully functional improvements or stabilization in correlation with drug treatment. Now, we see Becker muscular dystrophy as a secondary mitochondrial disorder, where the abnormalities in dystrophin initiate multiple stresses at the cellular level. Through our approach, we hope to use a stress adaptive response to allow the mitochondria to cope with disease-related stress. And this is all stemming from the fact that we have identified important parts of our mechanism of action within the last year. And we believe that our small molecule approach through this technology will allow us to restore the energy balance in the cells as well as regenerate muscle. Now, in their work to understand the biological effects of epicatechins, our founders realized that different catechins have very different biological properties. For example, in very early work, some epicatechins like plus catechin worsened injury in experimental models, while others like minus epicatechin had a protective effect. Now, this led our co-founders to believe that epicatechins were acting through a receptor-mediated pathway because structure appeared to be so important. Through computational modeling, they deduced that epicatechins were mimicking an endogenous steroid hormone, which itself has mitochondrial effects. They used in silico modeling, and that modeling suggested that a hormone called 11-beta-hydroxypregnenolone, which we call epindralol, was the steroid scaffold that epicatechins mimic. Now, our scientists were the first to describe this structure in this proposed role, and through subsequent research, we have been able to measure this hormone epindralol in human beings, and in animal models, we have shown that it actually increases with exercise. So this is a hormone that appears to help the body adapt to chronic stress, and exercise is a very good example of chronic stress. Our research shows that epicatechin and epindralol are associated with improvements in muscle and mitochondria. So structure is very important. And here you see the hormone in the middle. And here you see minus epicatechin, which is a weaker isoform of the drug that we studied in the past, and EPM01, which is what we're studying now. These are stereoisomers of each other, which means that you could almost superimpose them on top of each other, but not quite. They're almost mirror images of each other. Now, looking at epindralol in the middle, we see that EPM01, sorry about that, EPM01 has much more structural homology compared to minus epicatechin. And that's specifically related to these axial hydroxyl groups. And in our experimental models, we have seen that this difference in structural homology contributes to about a 400 times difference in biological activity, where EPM01 is much, much more biologically active than minus epicatechin. It also explains why plus catechin actually led to worsened heart injury in animal experiments. So we have chosen to develop EPM01 because of this profound difference in its biological activity. It is very difficult to purify and to manufacture. It is extremely rare in nature, but our scientists have found a way to manufacture it in a synthetic fashion in a very cost-effective manner. So we believe that functionally mimicking epindralol, it has important consequences. First, we believe that this leads to improvements in mitochondrial function as well as tissue regeneration. And so for patients with Becker muscular dystrophy, this could change the trajectory of their disease over the course of treatment. So why are mitochondria so important? Well, we know that they produce almost 100% of the energy that our cells in our bodies need. And that's very true, and it's very well documented. But they do more than that. Mitochondria also have other critical functions. For example, they direct the destruction of diseased or damaged tissue cells. They direct the destruction of unhealthy mitochondria. They are the source of hormone production. They help tissues adapt to stress and adjust to changes in their environment. Excuse me. And they communicate closely with the cell nucleus to regulate energy production and help cells adapt to changes. So what happens in dystrophinopathies like Becker or Duchenne muscular dystrophy? Well, the missing or abnormal dystrophin leads to abnormalities in the muscle cell membrane. Muscle cells and mitochondria are then subject to very large concentrations of calcium, which are toxic to mitochondria. These mitochondria stop producing energy, but unfortunately, they continue to produce damaging substances like reactive oxygen species, which in limited quantities, reactive oxygen species are helpful. Eventually, you end up with inflammation and fibrosis and cell death, and patients suffer changes to their physical function and quality of life. So we are developing drugs to try and stop or slow disease progression by improving the health of the mitochondria and also regenerating muscle. This is an important slide because it is early evidence of changes in muscle and mitochondria that the platform can achieve. This is work that was done by two of our early co-founders in 5 elderly patients with type 2 diabetes and chronic heart failure. They did not have Duchenne or Becker muscular dystrophy. And what you're seeing here is the result of 3 months of treatment with epicatechin on quadriceps muscle tissue. In the upper left, this is a pre-treatment image. You can see significant disruption of the sarcomeric structure, very small and abnormal looking mitochondria. In the upper right, this is representative of a post-treatment muscle biopsy where you can see much better looking muscular architecture and much more prominent mitochondria. Now, if we look further at the proteins obtained through these biopsies, we see several important things. First of all, we see increases in an important regulatory protein called PGC1 alpha. PGC1 alpha is a master regulator where it turns on or turns off other subsequent proteins. We see increases in proteins that are associated with mitochondrial biogenesis, including TFAM and mitophyllin. We see increases in electron transport chain complex 1, and this is ATP synthase complex 5. So this is telling us that there's an increase in mitochondrial mass. And then lastly, we see increases in the cristae. The cristae are the finger-like structures inside mitochondria that are associated with energy production, because on the cristae are where ATP synthesis occurs. So these are all going up in this study. Now, these Western blots are from the same trial, and the B lane is before treatment, and the A lane is after treatment. There are 5 patients, so you see 10 lanes in these Western blots. Inside the red box, we see that before treatment, these patients with diabetes and heart failure actually had a relative lack of dystrophin. It's almost like this advanced diabetes was a form of acquired dystrophinopathy. After treatment, you can see dramatic increases in the amount of dystrophin in these quadriceps muscle biopsies, and you can also see increases in the dystrophin-associated protein complex, including the sarcoglycans and dystroglycan. Also see increases in dysferlin, which is involved in muscle membrane repair. On the left side, we see important changes in regulatory proteins, increases in the level of follistatin, which is implicated in muscle regeneration, and reductions in myostatin. And then we see encouraging increases in proteins that are associated with differentiation from muscle stem cells down towards becoming muscle fibers. Now, these images are images of mitochondria from a study that we did with Professor Craig McDonald at UC Davis several years ago. Professor McDonald took highly purified minus epicatechin that we provided him and treated several patients with Becker muscular dystrophy for 2 months. And he performed biopsies of the biceps muscle before and after treatment. And what you're seeing here in this image is a mitochondrion. This is before treatment. These are cristae, and after treatment, what he and his co-investigators noticed was that there are more cristae present in the mitochondria, suggesting that the drug is associated with an increase in the energy-producing apparatus of the muscle cells. We also have, from this study, increases in dystrophin from the muscles of these patients. Dr. McDonald and his colleagues published most of the findings from the study in Muscle and Nerve late last year, and we expect a future publication to include the dystrophin data from this trial. I want to end with some cardiac data. As we've heard from Professor Figueroa earlier, heart involvement is an unfortunate consequence of dystrophinopathies. And so it's encouraging that we see this reduction, this trend towards a reduction in BNP from these patients. On the left, what you're looking at is BNP changes from the patients with type 2 diabetes and heart failure. and you can see a modest trend towards reduction. And that's not surprising because these patients did have heart failure, although there was no intervention made on their heart failure regimen during that study. On the right side, what you're looking at are BNP data from the patient's Professor McDonald's Becker study. And you can see here as well, there is a reduction after treatment with epicatechin over 8 weeks. Because BNP is an important prognostic biomarker in heart failure and levels over 100 picograms per milliliter can suggest significant heart disease, it's encouraging to see these data. We're working to understand why this is happening, and we're studying this very closely in our current Becker trial. So to conclude, our platform uses structural mimics of a newly described steroid hormone that appears to be associated with physiologic stress adaptation. We chose to develop EPM01 because of its structural similarity to this hormone, which appears to confer a much greater biological activity. We believe that modifying the disease course in Becker muscular dystrophy may require more than one biological strategy. For example, we think our platform couples bioenergetic improvement for the cell along with tissue regeneration and potentially other effects. We have seen consistent tissue and or plasma biomarker effects with the weaker isoform in diabetes and Becker muscular dystrophy, as well as with the current drug EPM01 in a prior trial of Duchenne muscular dystrophy that we did with Dr. McDonald. The possibility for cardiac effects is being examined further in our current trial, and that phase 1 trial is now nearly fully enrolled. We are continuing our research onto the potential applications of this platform in Duchenne muscular dystrophy. And I'll conclude by thanking all of our study volunteers and their families, and to our dedicated team of investigators and research staff. Thank you.
Grazie mille Ranzi per questa tua presentazione e adesso diamo la parola all'ultimo intervento previsto in questa mattinata, prima delle domande e risposte, ed è quello di Alan.
So, thank you very much. Now the floor goes to Alan Brussell from Edgewise Therapeutics. He is going to be the last speaker in this session. He's a co-founder of Edgewise, and today he will be dealing with the new project that may also regard BMD patients. You have the floor.
Thank you very much. Hopefully you can see this okay. And thank you so much to the previous speakers and Antonio for doing such a great job introducing the situation in Becker. Edgewise Therapeutics, we're trying something a little bit different for the therapy of muscular dystrophies. And I'll give you an early look as to what we're attempting in these important diseases. So we were launched in 2017, and our goal really is focused towards muscular dystrophies. And we have made a series of novel muscle modulators. I'll tell you about those.
Scusami, Alan.
Slides, with a focus on
Alan.
that we've made.
Alan, perdonami, la regia.
Sorry, Alan. There may be a sound problem. Can you maybe get closer to the microphone so we can hear you better? Otherwise, it's difficult for the interpreters to, hear you and translate.
Not a problem. Let me just try turning it up.
Grazie.
No problem at all. Okay. How's that? Good?
Sì, sembra di sì.
So we have made a novel compound, EDG5506, that I will tell you about, and we are currently in phase 1 clinical trials in healthy volunteers with a goal towards starting studies in Becker muscular dystrophy in 2021, later this year, and in Duchenne next year. We're founded by a number of pharmaceutical company veterans, and I just wanted to point out one person that you may know quite well, Abby Bronson, who is listening into the call today, is a well-known member of the PPMD organization in the United States, and we're really honored to have her as part of the team to help coordinate our studies with patients. We also have the usual kind of high quality set of scientific advisors. John Vissing, many of you may know as the coordinator of the World Muscle Society meeting last year. Lee Sweeney has been in the area for many years, and we also have Barry Byrne and Leslie Linewand as great references. We also have a number of contacts with the patient advocacy organizations, including PPMD, Cure Duchenne, Treat NMD. So I don't need to focus too much on the role of dystrophin in muscle health, but we do have a different focus. So I will give you some idea of that. So we see the primary role as of the dystrophin complex as a connector between the extracellular matrix of muscle and the muscle contractile machinery. So it spans the membrane and it connects those two important parts of muscle, and that really aids the integrity of skeletal muscle. When muscle contracts, then what you can see is this internal stress that eventually leads to hypercontraction and stress and collapse. And that's largely driven, as was mentioned before, by changes in membrane permeability, calcium entry, and then hypercontraction. I'm going to focus on this a little bit more over the next couple of slides. Here is that histological information on the previous slide as a figure. Here's the dystrophin complex spanning the extracellular main all the way into the contractile machinery. In the normal state, when muscle contracts, the dystrophin connects the contractile machinery to the extracellular matrix and allows the membrane to flex when the muscle contracts. If dystrophin is not present, or it's truncated or mutated, then that inherent connection is lost. So when the muscle contracts, you get a discordance between the contractile machinery and the membrane, and that results in this sarcomeric collapse. And you can see it here, and I will show you that in even greater detail on the next slide. This is one of my favorite papers for Duchenne, published back in 2008, and it actually is a movie of dystrophic muscle from the mouse contracting. So these are researchers in Michigan. They use a very small muscle from the finger muscles of the mouse, and it's so small you can see through it with phase microscopy. And as you can see, when this muscle, the dystrophic muscle, is contracting, the membrane stress that's resulting from this discordance drives calcium entry and hypercontraction of these fibers. And you can actually see them slithering around because they've been proteolytically cleaved by calcium-dependent enzymes. And this, of course, is what goes on in Becker and Duchenne muscular dystrophy muscles every day. It really is the result or is the cause of the stress and the inflammation that eventually leads to the fibrosis and fat accumulation that were discussed in the last few talks. So we have come up with a novel strategy to try and avoid that collapse occurring. What we found is in the literature and with our own research that skeletal muscle has some muscle fibers that are more susceptible to injury than others. We're made up of fast and slow skeletal muscle fibers. And what we found is that fast skeletal muscle fibers degrade with a greater frequency than slow muscle fibers. We've made an investigational therapy, 5506, that selectively prevents the degradation of those fibers by reducing the force of their contraction. It's a counterintuitive hypothesis, but I'll show you a little bit of data to show how exciting that may be. In animals, what we found, doing studies in mice and dogs with muscular dystrophy, is that EDG5506 protects these susceptible muscle fibers, improves the muscle fiber function, and reduces fibrosis at doses that don't inhibit integrative strength or other kind of activities. Now, of course, these are all preclinical data. In humans, we, of course, don't know yet whether those translate into positive effects, and that's why we're doing the clinical studies that we've initiated now. Just a little focus upon the contraction of muscle and physical function. So, of course, if you've ever had a muscle cramp, you know that maximal contraction of muscle is not productive. Your muscle tends to kind of ball up. It's very painful and not productive to motion. The target goal for 5506 is to use our pharmacological strategy to decrease the contraction of muscle between 5 and 20%. So we're just taking away the hypercontraction, but leaving the functional contraction of muscle. At this level of inhibition, what we've seen in the preclinical models is the animals retain their strength, but then the muscle degradation is dramatically attenuated. I'm just going to show you one data slide. We have a publication in submission that hopefully you'll all be able to see fairly soon. I'm just going to show you one data slide, and this is from dystrophic dogs. So we have a lot of data in dystrophic mice, but of course, the mouse is a long way away from the muscle of a human. And as such, the dystrophic dog, in this case, the golden retriever with muscular dystrophy, is much closer to the humans. Their muscle composition is quite similar, and the speed of degradation of that tissue is quite analogous to humans. So what we did is we dosed older dystrophic dogs. So these would approximate to a 10-plus-year-old Duchenne patient with the compound as an oral gavage in 2-week dosing blocks. We dosed vehicle or placebo to active drug and then back to placebo. And we measured two things. One is we measured circulating CK. Of course, that's higher in dystrophy. And we saw a 60% decrease over the dosing period in CK. And then what we used is an activity monitor that was attached to the collar of the dogs just to measure a habitual activity. And we saw a 30% increase in habitual activity. So we decreased injury and we increased habitual activity in 2 weeks of dosing. So unlike many other approaches, ours is not a gene therapy. We're a small molecule approach that's designed to protect skeletal muscles in conditions where there's weakness associated with structural differences. What we have is a once-a-day tablet or suspension. We're going to protect those susceptible muscle fibers from degeneration. In preclinical studies, we've seen protection of both skeletal and cardiac muscle, which of course is a very important observation for both Becker and Duchenne muscular dystrophy. It's appropriate for any age or mutation. Of course, as patients get older, our dosing approach will have to change because of the amount of residual muscle that's left. But as such, our approach is appropriate for Duchenne, Becker, limb girdle, and potentially even metabolic myopathies. Where we work in the disease cascade is right at the top here. So we stabilize that membrane by binding internally into the muscle to limit the ability of contraction to lead to stress. As such, we're right at the top of the cascade. We're not replacing dystrophin. We're dystrophin mutation agnostic. We're essentially an orthogonal strategy to protect the skeletal muscle in a variety of very important diseases. We should be combinable with any developing approach because we're really a very different type of strategy. So whether that's glucocorticoids or other gene therapy approaches, we should be combinable. And because we're at the top of the disease cascade, we hope that we can arrest all of these events, the inflammation, regeneration issues, the stem cell exhaustion, and the fibrosis that really eventually limits physical function. So where we are on the therapeutic cascade is right now we're doing single and repeat dose studies in healthy volunteers in a first time in human test center in the United States. Starting around the summer, of this year, we'll be recruiting Becker muscular dystrophy patients to the first time in human test center to do 2-week repeat dose studies just to look at tolerability and biomarkers. So this isn't really a long-term efficacy study. This is a very focused study just to compare drug exposures in Becker patients to those in healthy volunteers. After that, we'll be running a separate Becker proof of concept study. This will be a 1-year study, very similar to the Imperium design that you heard earlier. And we will be actively recruiting patients for both this study and the United States study now. And then the goal is to initiate Duchenne muscular dystrophy studies in 2022. So thanks very much for your time. It's kind of a novel approach, which often takes time to kind of understand and really acknowledge the kind of nuances of it. So hopefully we'll be talking in the near future.
Gerardo. Grazie mille Alan, do la parola brevemente a Gerardo per salutare i relatori.
Sì, grazie a tutti gli oratori. Iniziamo la
I wish to thank all the speakers, and now we start a Q&A. I don't know. I immediately answer the first and second question. There is a dad following us from Greece and is asking us whether guidelines will be available in English. The answer is yes. We'll be working on it. We are translating them so that they can be accessible to all patients, so not only Italian patients. He's the father of an 11-year-old boy, and he has a good state of health, even without taking drugs. How can we be sure of it being a Becker and BMD? And please, can we have on the video the other clinicians, because we know that there's Dr. Ferlini. There are many of you who can also take the floor to answer this and other questions. Harris, how can we be sure it is a BMD?
Se non risponde nessuno.
If no one is answering, Elena, would you like to step in? 45-47 deletion is a classical Becker deletion. If the molecular diagnosis is correct, the diagnosis is correct. It is very well known as a type of mutation. It's not a rare mutation. It is a frequent mutation in patients with BMD. Giacomo, if you want to add something. Well, the validation criteria for mutation, among the criteria, we always have the recurrence within a given disease, and the 45-47 deletion is the most common mutation in Becker BMD type dystrophinopathies. You have had this diagnosis very early, so this father wants to know what is the prognosis and what is going to happen in future years to this boy. Of course, this is a more difficult topic because we can foresee the evolution, a classical evolution of BMD, but the boy must be followed according to the existing standards of care that are precisely those that are described. And then we have Gianfranco, who has a BMD, she is a BMD patient, age 50. She still walks. She has a nonsense mutation in exon 37. And the question is, what is the research underway? Gianfranco, I mean, what you heard today are what the trials that are going on for Becker. is there any other study? But I think we have given a good overview of scientific research in trials on BMD. Another question by Dr. D'Amico for Professor Comi. She's asking if you have noted alterations in thyroid hormone. Thank you. The answer is no, we didn't. We have a side study on the lipid profile of adults in the treatment with givinostat, induced by a number of observations pertaining to this trial. Now we are waiting for the results. And one more general consideration that applies to all trials, studying a pediatric population as in DMD, or a population that ranges from 18 to 70 years of age, as in the case of BMD, because adults have problems of coexistent diseases that are typical of adults. So it is a universe, really, where we must consider a number of variables very different from children. In a DMD trial, we have observed high levels of triglycerides. We had not observed it in the pilot study, whereas on larger numbers, on larger series, many patients had this problem. I was asking about thyroid hormones because in the pediatric population we had observed a resistance to TSH. I don't know whether this may be linked to lipid impairment. Thank you. We are a bit late, and we close with the last question. Well, can we have something from you in Italy as well? Of course, we are waiting for you in Italy as well. In Epirium, the question is for the Epirium Bio, for Ranci Samara.
Okay.
Possible clinical studies once you have completed those that are underway. Are you considering going outside the United States? So in Italy, and more specifically in Europe and in Italy?
Yes.
And one final question for Edgewise. Is your molecule similar to TPAs?
No. It's a small molecule modulator of muscle contraction, but it is not similar to an ATPase.
Grazie mille.
Our next trial, we will be doing a global trial, so we will definitely be looking for participants from Europe.
Grazie mille anche a te.
Thank you very much indeed. And since it's late, it's very late, it's lunchtime. So we wish to thank all the speakers who were with us today, as well as all those who contributed to drawing the guidelines. Well, we reconvene the session at 3 p.m. with a beautiful session on the psychological aspects of adult life, where, of course, we give the floor to patients. So please follow this session also from abroad. That as well is going to be translated, so you can listen to it in English. And I wish to remind once again our Easter campaign. You can order our Easter doves that can fly even outside of Italy. You can write an email and you get this cake at home. And well, enjoy your lunch, and thank you very much indeed.
Grazie mille.
Grazie.
Ciao a tutti.